Hello everyone, welcome you all. Okay, so we have been completing this chapter and it's almost complete, and today is the last class of this chapter, Cell the Unit of Life. So what we'll be studying today is about very important thing, but in NCERT if you think they have given very only 2 lines about that. So you people might feel it so much we need to study and only 2 lines are given in NCERT, and you know very well, always I'll be telling you that whatever is given in NCERT, yes, obviously it is we have to do that, but still to understand those 2 lines also we need to understand something, right? So today what we'll be doing, and today will be the last class of this chapter. After this what we'll do is we are going to solve the questions, okay, of previous year questions we'll be solving. And one more thing I would like to tell you, after this class when you come to the next video, you need to be completely prepared with the chapter. Then only you are going to come to the next class and you're going to watch that video and you have to answer yourself as well. So please be prepared with this, okay? And the last class today, we are going to talk about micro bodies. Micro bodies. Okay. Now, what are micro bodies? See, micro bodies, name itself you'll understand, right? Micro bodies, what? Micro means what? They are very minute, you can say. They are minute structures, they are bodies, somas. So they are minute bodies which are present in the cell. This is one thing. I hope this much is clear now. Minute bodies means what? In comparative to other organelles, these organelles are very, very small in size. That is why we name it as micro bodies. Then what we need to study about this? About minute bodies. We can call them as microsomes. And why zomes? Why soma? You know what is soma? Soma means we use it for bodies. So microsomes means minute bodies. We can also call it as microsomes, and we can also call it as cytosomes. In exam, if anything is asked regarding microsome or cytosome, remember that is nothing but they are asking about micro bodies. Hope so this much is clear for you people. Okay. Now, where do you find it? You will find it in the cytoplasm of the cell. And which microbodies we are talking about? Prokaryotic or eukaryotic? Remember one thing, microbodies are present in eukaryotes. They are not present in prokaryotes. So we are talking about microbodies of eukaryotic cell. That means in both plant as well as animal cell, we have microbodies and they have some particular function that we are going to see that. Okay, which are the microbodies, which are the types as well. Take down this much. Okay, fine. So now coming to how they are produced. They are very minute. In NCERT they have given that they're just minute vesicles. They are just like vesicles. When you heard this term vesicles, what do you remember? What do you remember? You remember that when we were doing about cell, this was the nucleus. Surrounding the nucleus, we had this structure like this, endoplasmic reticulum, right? Endoplasmic reticulum. And if it is rough endoplasmic reticulum, it will have what? Obviously, if it is rough, it will have ribosomes on its surface, right? Like this. And smooth will not have ribosomes. So this is what? This is rough endoplasmic reticulum, RER. And what about this? This is smooth endoplasmic reticulum. Again and again, why I'll be repeating? Because you know whatever I told you, many times I told you. So we need to be prepared, okay, for exams. The most important thing for the preparation is not just understanding, it's revising. Again and again the things should be moving in your brain, then only you'll understand. So always I'll be doing the same thing again and again so that it should be fixed in your mind, okay? Anything which we practice more, it becomes more permanent in your head. So this is rough endoplasmic, this is smooth endoplasmic. What is in the center? This is the nucleus, right? Okay, now rough endoplasmic reticulum, if you see the structure in outside, so in textbooks as well as what in your books, if you have seen, you will find that there are some vesicles which come out from this structure like this. There are some vesicles which come out from this. Have you observed that? They will be showing these vesicles, and we have detailedly talked about vesicles when we were doing which concept, that is endomembrane system, in this chapter itself starting, right? So there we have told that vesicles, if it is rough endoplasmic reticulum, then it will be obviously producing what? Proteins. Why? Because it has ribosome on its structure. So those ribosomes, it will produce protein, and that protein, if it has to be sent out to the Golgi complex, it is sent in the form of vesicles. Proteins will get embedded into the vesicles, and vesicle will form a small structure, and then vesicles move into the cytoplasm. So these vesicles, we are talking about vesicles, these are very small, and these vesicles itself, we call them microbodies. What do we call them? Microbodies. Why? Because they are very minute in structure. Then what is the importance of these microbodies? How they function? What is their importance? So their importance, if you are talking about, now they are still called as microbodies, but when they have some particular enzymes for their function, then different names are given. Okay. Now what will happen here? These vesicles have some proteins within them, and that proteins, along with proteins, these vesicles will come out into the cell cytoplasm. Now for example, these are the vesicles like this. Don't get confused with ribosome. Ribosomes are in green. Red are vesicles. So these vesicles, even endoplasmic reticulum produce vesicles. Smooth also will produce vesicles. Golgi bodies also produce vesicles. Like that only, lysosomes are formed from Golgi bodies. In this form only, like this only, right? So that you know very well. Now vesicles, they start moving into the cytoplasm. Then what will happen? What these vesicles have within them? They have proteins within them. Why? Because if they are produced from rough endoplasmic reticulum, then obviously proteins are there in them. Then when they are moving in the cytoplasm, so many other proteins will get embedded into it. Okay, and then it forms particular microbody based on its function. There are different types of microbodies. Okay, now they'll be moving in the cytoplasm. They have so many functions to do. Okay, now what is the peculiar thing about microbodies? Single lipid bilayer. What it has? It has single lipid bilayer. This is just an example I gave you. So many microbodies are there based on different functions. So what I'm trying to explain you here is that how it is produced and how it starts moving in the cytoplasm. Okay, so I think this much is the crude thing what you have understood here. Now let us see. So they are made up of single lipid bilayer. They have only single layer of lipid. Lipid bilayer, no? Like chloroplast has two membranes, nucleus have two membranes, mitochondria has two bilayers, two membranes. Likewise, it has only single membrane. And so many proteins, and they form in the, they function as enzymes within them. Okay, they have single lipid bilayer, which is very, very important for you to remember. Okay. Then what else? Now let us talk about types, different types of microbodies. In NCERT, nothing is given about types, but you people should know these types. Okay, we will talk about types now. The first type we are talking about is, you know very well about this. We call it as peroxisomes. These are the one type. Then we have glyoxysomes. Then we have spirosomes. And then we have lomasomes. Please take down this much. Which are the types? Which are the types? So we have different types here. Types of microbodies: peroxisomes, glyoxysomes, spirosomes, and lomasomes. So what you need to remember here in this is, we are going to talk in detail about everything, but peroxisomes are present in both plant as well as animal cell. Because these are eukaryotes, no? Then obviously they are present in both plant and animal cell. Glyoxysomes, peculiar thing is, they are present only in plant cell. And these are also present only in plant cell. And even these are present in plant cell. These three are not present in animal cell. These are also found in some fungal cell as well. Okay, but what you need to remember is only microbody which is present within both animal cell as well as plant cell is the peroxisomes. And very well, we have talked about peroxisomes when we were dealing with chapter photosynthesis in higher plants. Hope you people remember. Okay, anyways, even if you don't remember, we're going to talk about again about these things. Take down please. Peroxisomes, glyoxysomes, spirosomes and lomasomes. Based on their function, there are different types of microbodies. Clear? Now, one more thing, I'll clear it now only. This glyoxysomes, they are not a different microbody. They are peroxisomes only. But based on their function, we call them specialized peroxisomes. Peroxisome, they function in a different way to form glyoxysomes. Sometimes in case of plant cells, okay? Anyways, we will talk about that now in detail. So the first microbody we are talking about is its peroxisomes. Fine. So the first one is peroxisome. So just we are not going in detail. We are not going in detail because however it's not given in detail in book, at least in your NCERT at least. So we will not waste our time going in detail for everything. We will just know what it is. Okay. Now coming to peroxisomes, I told you these are present both in plant as well as animal cell. Where do you find them? You find them in plant and animal cell also. Right? Okay. Now we will see in separate sense like what is their function. But it has a common function. Even in plant as well as animal cell, it has common function. And why do we name it as peroxisome? That means it has some function with that. That is, it helps in synthesizing, or you can say producing. Anyways, we can use the term as, it also, you can say, it produces and destroys. Both is done by this only. What is produced? What is destroyed? It produces and destroys H2O2. What is H2O2? Sorry, it's H2O2 is called as hydrogen peroxide. Alva? H2O2 kenanta karitivi? What do we call it as? H2O2 is known as hydrogen peroxide. So what is the function of peroxisome? It produces H2O2. Also it destroys H2O2. Confusing, right? Then why it produces if it wants to destroy? This question comes to your mind. And do you remember where you have studied this? In photosynthesis chapter. Do you remember? We will see that. So it helps in what? It helps in producing H2O2. Also it helps in destroying H2O2. Then why it produces if it wants to destroy, right? We will see that now. Okay. So this peroxisome, I'll tell you, it is very, very important for animal as well as plant cell. Why? Because it helps in detoxification. Now for example, hepatic portal system, right? In case of school, when you were studying 9th and 10th standard, you might have heard about this hepatic portal system. Because whatever we eat after digestion, the blood, after absorption of the minerals and whatever the digested food, it will not directly go to heart. Instead, it goes to liver. Because their liver, what it does, it checks out what is poisonous in the blood, if anything toxic is present in the blood or no. That checking is important. If it is not checked, then that blood is directly moved to heart. And heart will pump that toxic blood to all the parts of the body. Then every toxic material is passed on to every cell of the body. Then that becomes very dangerous. So this checking is done in liver, in the animal cell, right? In animals, in humans if you talk about, and in animals. So different various functions are there, but to understand we will see it helps in detoxification, where peroxisomes, they are found in kidney cells and they are found in liver cells in more number in case of animal cells. Okay, first we are talking about animal. Now I'll tell you what it does. See, what are the enzymes which are present in peroxisomes? Which are the enzymes present? I told you they are single layer. For example, this is a single layer, and what it has inside? It has various enzymes present inside. Peroxisome has various enzymes present inside. Now among these enzymes, we are talking about different enzymes which are present here. So first enzyme which is present here is oxidase enzyme. Then we have catalase enzyme. Then we have one more, that is urease, that also come under oxidase. Anyways, we have luciferase enzyme. These are the different enzymes present in peroxisome. Okay, what it does? What it does? So we'll talk about that. See, what does oxidase will do? Oxidase enzyme help in oxidation of amino acids and uric acids which are present, which are produced in the body. What it does? This oxidase. Okay, you have taken down this much. Please stop the video and take down if you want. Okay, then we are going to go further. So what is done by this peroxisome enzymes? I'll tell you what is done. Now ammonia, or sorry, amino acids and uric acids. These acids, you know, they are toxic to the body. We need to remove it out from uric acids are very dangerous for the body. We remove it out from excretory system. So likewise, what this peroxisome will do is, with the help of this oxidase enzyme, it will oxidize this amino acid and uric acid. Then what is the byproduct which is produced is H2O2 is a byproduct. In total, I am trying to explain you. Okay, we are not going in detail chemistry here. See, amino acids and uric acids are toxic to the body. We need to, we need to destroy it, or you can say we need to digest it, or we need to do something so that it becomes less toxic. So what we do is we oxidize it with the help of peroxisome. How? It has oxidase enzyme. Oxidase will convert this amino acid and uric acid, it oxidizes it. But what is the byproduct produced? The byproduct is H2O2. Now, do you think H2O2 is good for the body? No. H2O2 is called what? It's hydrogen peroxide here. What is H2O2? H2O2 is hydrogen peroxide, right? This is a byproduct which is produced when we oxidize amino acid and uric acid. But even H2O2 is toxic. So now what this peroxisome will do? That only produced H2O2. Peroxisome only produced H2O2 while oxidizing amino acid and uric acid. Now what it will do? This H2O2, which is toxic to the body, with the help of enzyme. Now which enzyme it will use? It will use this catalase enzyme, and using catalase enzyme, what it will do? It will catalyze this H2O2 and forms H2O plus half oxygen. Are you understanding what I am trying to say? Please listen carefully. What happened here? Amino acids and uric acids, finally, they are not good for the body. So it has been oxidized with the help of peroxisome, with the help of enzymes which are present in peroxisome like oxidase. What is produced as a byproduct? H2O2, which is called hydrogen peroxide. It is a byproduct. Even this H2O2 is not good for the body. Even it is toxic. So now what peroxisomes will do? It has one more enzyme called catalase that will catalyze H2O2 and forms water and oxygen. Now 2 times if it does that, 2 molecules of water and 1 oxygen can be, produced in the body, which is good for body, right? That is why I told you it is called peroxisomes, because it helps in synthesizing and destroying hydrogen peroxide. That is why the name is peroxisomes. Clear? So this is the work of peroxisome in case of animal cells. Fine. Now what it does in plant cells? What is its function in plant cells? That we need to study, right? So this is, I think you understood this in a clear sense. It also has urease enzyme. It helps in catabolism of uric acids. It has luciferase enzyme also. You know what is that luciferase enzyme? This is just for your information. Luciferase enzyme. Have you seen this fireflies? Night time. Have you seen this fireflies what is that? It produces light. Nodirbekkalla light idaguttalla, illuminate aguttalla ಕೆಲವು organisms during night time ಕೆಲವು ಹುಳ ಇರುತ್ತೆ, ಏನು ಅಂತ ಕರಿತೀವಿ ನಾವು ಅದಕ್ಕೆ, light produce ಮಾಡುತ್ತದು, I mean light ಕಾಣಿಸುತ್ತೆ ಅದರಲ್ಲಿ. So that enzyme which is present there to produce light in those organisms, it is luciferase enzyme. So it is a it's an enzyme of peroxisomes. Okay, so these are the function of enzymes of peroxisomes. Now, function of peroxisomes in case of plant cell. ಇದೆಲ್ಲ animals ಬಗ್ಗೆ ಆಯ್ತಪ್ಪ. Okay, then ಹಾಗಿದ್ರೆ plant cell ಅಲ್ಲಿ peroxisome function ಏನು, what it functions, how it functions in case of plant cells. So plant cell ಅಲ್ಲೂ ಕೂಡ function same ಇರುತ್ತೆ. Hydrogen peroxide ಎಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಿದ್ದೇವೆ? Do you remember? In plants, peroxisome function in plants. We have studied this very important concept of photorespiration. Do you remember that? Photorespiration NPD allargo. What we had done in that in this concept, what we have studied, photosynthesis is a good thing for plants. It produces glucose with the help of light reaction and dark reaction of photosynthesis. But this is a ulta process. Ali in photosynthesis, RUBP, it what you can say, RUBP, it combines with carbon dioxide to form 3-PGA, alva. But here in photorespiration, instead of combining with carbon dioxide, RUBP in madutte, it combines with oxygen and it forms phosphoglycolate. If you don't remember, please turn the pages back and revise and then learn it again. So phosphoglycolate, adu along with PGA produce agutte, alva, there in photorespiration process. Then that phosphoglycolate will form glycolate in chloroplast. Avage ಏನು ಸ್ಟಡಿ ಮಾಡಿದ್ವಿ ಪ್ಲಾಂಟ್ ಸೆಲ್ ಅಲ್ಲಿ in photorespiration study ಮಾಡಬೇಕಾದ್ರೆ 3 organelles we have studied. First one was chloroplast. Chloroplast ಜೊತೆ ಏನಿರುತ್ತೆ? Next we studied this peroxisomes. And then we studied one more. ಇವು ಮೂರು ಕೂಡಿ ಕೆಲಸ ಮಾಡುತ್ತೆ ಅಲ್ವಾ? Mitochondria. Do you remember that? So chloroplast ಅಲ್ಲಿ phosphoglycolate form ಆಗಿದ್ದು glycolate ಆಗುತ್ತೆ. And the glycolate enters peroxisome. So peroxisome ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? Glycolate is converted into glycine. Do you remember this? ಈ process ಆಗಬೇಕಾದ್ರೆ there is H2O2 released, which is hydrogen peroxide. Again catalase enzyme ಇದನ್ನ ಮತ್ತೆ water ಮಾಡೇ ಮಾಡುತ್ತೆ. That is not a big deal. Here we are talking about this cycle, photorespiration. What do we call it as? Photorespiration. ಇದಕ್ಕೆ ಮತ್ತೊಂದು ಹೆಸರಿದೆಯಲ್ಲ? What is the other name for photorespiration? I want you all to comment in the comment box to find out whether you people still remember it or no. Which cycle? We also call it as one more name is there. Fine. So glycolate is converted into glycine in peroxisome and glycine will enter mitochondria. Mitochondria ನಲ್ಲಿ glycine ಏನಾಗುತ್ತೆ? Serine ಆಗುತ್ತೆ. Serine will come back to peroxisome again. Do you remember this? And serine will convert into what? Serine is converted into glycerate. And then glycerate is combining, it also again forms RUBP and the cycle continues. This is a wasteful process in plants. And which plants photorespiration will occur in? ಯಾವ plants alagathri idu? C3 plants. You will not find this photorespiration in C4. Why you will not find that also you will comment it. Okay. So this is all about peroxisome. This is how it helps in plant cells. Okay. So okay, this much you just know it and just take down. Peroxisome is very much important, and for you people peroxisome jothe glyoxysome kuda tumba important. Okay, iveradunu detail alli madana. I mean after that the other things if you know little bit it's okay. So and the most important one is peroxisome only. So please note down everything.